Hello and welcome. My name is Claire Brummitt. I'm here today to talk about my capstone project, Smoking Cessation CYP2A6 Inhibition by Trans-Cinnamic Aldehyde. My capstone mentor is Dr. Janine Chan. So first, I'd like to talk a little bit about some background information about why research into smoking cessation, AIDS, and smoking in general is still really important. So first, over 34 million people in the United States are currently smokers. It's also the leading cause of preventable disease in the United States and costs over $300 billion each year. So, if smoking is so bad for our health, why do people continue to smoke? Well, it's because nicotine is an incredibly addictive substance. In fact, only 7.5% of smokers successfully quit in 2018, according to the CDC. To help us understand why nicotine addiction is so difficult to kick and how it works, we have to go into a little bit of the biochemistry. So, first, cytochromes, which is a superfamily of enzymes found predominantly in the liver, but all over the body, are responsible for metabolizing exogenous compounds, which are compounds not produced or natural to the body. Cytochrome P450-2A6, which is a submember of the cytochrome superfamily, is one specific enzyme that's actually responsible for metabolizing 70 to 80% of the nicotine in the body. So, by targeting CYP2A6, as it's known, it's been found that decreasing the activity of this enzyme lowers rates of lung cancer and lowers rates of addiction in people. So that leads us to the project purpose. The purpose of this project is to find and test TCA, trans-cinnamic aldehyde, on CYP2A6 alleles to ensure that it's actually an effective smoking cessation aid for a broad range of people. So, we do this through genetic engineering techniques to mimic naturally occurring enzyme or allele variants in the human population to ensure that TCA is actually effective on many different alleles. So, what's been previously done on the project? As I mentioned, trans-cinnamic aldehyde has already been found to be a CYP2A6 inhibitor, and it most likely acts to degrade the heme functional group, which is what allows it to break down nicotine and other exogenous compounds. This notation here with the star, this just denotes a specific allele that's been talked about in the literature. So they're given these names, so it's easy to, keep track of them. So previously on this project, star 43, star 23, star 17, and star 7 have been produced and explored. Specifically, star 23 was found to have overall lower activity, but it was actually inhibited at approximately the same rate as the naturally occurring star 1 or most common allele, showing that TCA is effective for that allele. And this is the goal of my project, is to test this, whether or not TCA is effective on star 11, which is my chosen allele. So some more information on star 11, which again is the allele that I chose. It's a naturally occurring rare allele that's found in Asian populations. In this specific mutation, thymine 670 in the genetic code is converted to a cytosine, and this leads to a substitution missense mutation where amino acid 224 is converted from serine to proline. And this enzyme overall shows lower activity, and that's actually how it was discovered here on another exogenous compound that CYP2A6 metabolizes. It's been found that star 11, which is shown here in the black, has overall lower activity compared to the natural wild type here. So, once I've chosen a mutation, how do we produce it? We produce it through site-directed mutagenesis, which is described here. This is a process in which a section of the DNA where you want the mutation to be introduced, you produce a complementary sequence known as a primer, which is just a small chunk of complementary DNA which contains the complement to the mutation you want to introduce. So that's a little confusing. Let's look over here where I've given an example. This is actually a primer that I used. This is the initial sequence in the DNA that codes for the CYP2A6. This is the thymine that we want to convert, thymine 670, that we want to convert to cytosine. So my primer sequence, which is complementary again to this sequence, in it you'll notice this is the highlighted complementary mutation. So I want to introduce a cytosine where this thymine is. So I put the complement of what I want to introduce in this place. So guanine is the complement to cytosine, so I put a G here, and it will replace the T with a C when we continue with the site-directed mutagenesis. So the primer, because it's very specific, it anneals preferentially to the site where we want to introduce the mutation. It anneals or binds complementarily to that sequence of DNA. And then a polymerase, which is another enzyme that's naturally found in the cell, can come attached to the primer and begin replicating the DNA. But when it replicates the DNA, it now integrates the mutation, this G here, it'll integrate the complement of the mutation of the primer into the sequence of DNA. So it'll introduce the cytosine we want. And from there, we use PCR to amplify the mutated DNA to produce a much higher concentration, so we can then send it off for sequencing to confirm that the mutation we desire has been introduced. So we had the primers synthesized for us at a lab. We performed the site-directed mutagenesis, and then we were using PCR to amplify the proteins. But then how do we ensure that the protein, the plasmid, was actually produced? We do this through gel electrophoresis, which uses molecular weight, or the size, to separate things, specifically DNA, by size and by charge. So bigger things travel a lot less. Shorter things travel a lot farther. That's what this ladder shows. Small, not heavy molecules travel much faster through the gel. Heavy things travel much slower, and so they don't go as far. In this first round, we actually got little to no product formation. We only had plasmid product seen in the 500 picogram DNA template tube. This is most likely due to the fact that we were using relatively old template DNA dilutions, and so probably the DNA was beginning to degrade a little in the freezer. It wasn't high quality anymore, so it had low amplification during the PCR. So we didn't feel super confident, so we wanted to continue to optimize the PCR and make it better. So we did this by optimizing the magnesium concentration. DNA is a molecule, has a backbone of very negatively charged phosphate groups. These phosphate groups, they don't like to be near each other because like charges repel. And so to stabilize this, you can actually add magnesium, which is a positively charged ion. It can complex with the negatively charged backbone and help to stabilize the DNA molecule and prevent it from falling apart due to charge repulsion. So we experimented with this, and we didn't see a strong correlation between increasing the magnesium concentration and improved PCR amplification. However, we did see a lot more product overall this time. As you can see, every one of the tubes had product in it of the correct weight. And this is, again, most likely because I was using new DNA dilutions, template DNA dilutions, which probably helped to improve the overall efficacy of the PCR reaction. And here in the 1.25 millimolar MgCl2 tube, you can see that there is product, but there's a lot less product when compared to the other tubes tested, and this is probably due to a pipetting error. Working in very small microliter, 10, 5 even microliter range, it can be very difficult, and this is again probably due to a pipetting error, but this would be something I would want to explore further to ensure that it is due to an error and not due to some other cause. So what are the next steps for my project? The next step is to perform the DNA purification off of the samples that I ran in the gel electrophoresis to purify the plasmids, and then perform a ligation reaction to prepare them for sequencing. We would then send them to a lab to be sequenced for us to confirm if the mutation we desired was incorporated into the plasmid successfully. And then from there, we would look to transform E. coli, competent E. coli with the mutant plasmid to have them produce the mutant CYP2A6*11 for us for then protein purification and testing with TCA. So some acknowledgements, definitely want to acknowledge Emily E. Scott. She provided us with the PKK2A6DH plasmid, which is the CYP2A6 plasmid that we used in this experiment. Dr. Janine Chan, obviously, my capstone advisor. I couldn't have done it without her. And Dr. Cortez for being a great resource for me and for pushing me to follow my passion in science. I wouldn't be here without all of these people. So thank you very much. And if there are any questions, I would happily field them.